Hello and welcome to CFTR Protein Defect Red Table Talk. I am your host, Josiah. Today, I will serve as the mediator for the discussion between our 6 respective CFTR protein defects. First, a little about their work. CFTR proteins are present in all mucous membranes across the body. However, today we'll focus on its role in the respiratory epithelium. The epithelium of your respiratory system is made up of mainly 3 types of cells: basal cells, ciliated cells, and goblet cells, respectively. Finally, we have the ciliated cell, whose membrane the CFTR protein calls home. Basal cells are a type of respiratory stem cell, meaning that they have not fully differentiated. If a ciliated cell or goblet cell were to die or needed to be replaced, a basal cell could differentiate to fill the role of one of these types of cells. Goblet cells are dedicated to the secretion of mucin. They have an extensive endomembrane system dedicated to pushing out the mucin protein, which, when combined with water, forms mucus, which covers all mucous membranes across the body. The apical membrane of the ciliated cell is covered in projections called cilia. These projections enable the ciliated cell to play an important role in the way pathogens and particulate matter that enter the respiratory tract are dealt with. These cells are able to move mucus, which traps pathogens and particulate matter, towards the esophagus, where it can be sent to the stomach and digested. This is called the mucociliary escalator. In addition to their role in moving mucus, ciliated cells also provide the water necessary for the creation of mucus. The apical membrane of the ciliated cell is lined with 2 types of transmembrane proteins, among others. The first, of course, is aquaporins, a proteins which enable water to move across the membrane that it normally would not able to, as water is a charged polar molecule. The second protein is the CFTR, or the cystic fibrosis transmembrane conductance regulator. This protein is responsible for the transport of chloride ions out of the cell. Normally, CFTR proteins pump chloride ions out of the cell, creating a hypertonic environment in the lumen. This encourages the movement of water out of the cell, as there is a higher concentration of solute outside of it. Here, the water is able to mix with the mucin and make mucus. In cystic fibrosis, however, this is not the case. Cystic fibrosis, commonly abbreviated as CF, is an inherited disorder that causes severe damage to the lungs, digestive system, and other organs of the body. Cystic fibrosis affects all cells that produce mucus, sweat, and digestive juices. These secreted fluids are normally thin and slippery. However, with CS, these fluids are dehydrated and become thick and unmoving, which interferes with the normal function of organ systems that rely on mucous membranes. The issue of dehydrated mucous membranes associated with CF is caused by defects in the CFTR protein. Furthermore, they are exacerbated by the ENaC, which allows sodium ions to flow into the cytosol of epithelial cells. This increased concentration of ions within the cells will draw water in, further dehydrating the mucus. Dehydrated mucus can cause blockages in the trachea, bronchi, and bronchioles, interrupting airflow and preventing gas exchange in the lungs. This can be life-threatening. On a more light note, let's introduce our CF-causing guests today. Hi, I'm the complete absence of a CFTR protein, meaning I cause the most severe cases of CF. I am most often caused by an early stop codon in the gene for the CFTR protein, resulting in an unstable truncated RNA that cannot be translated into a protein. Without a CFTR protein, no chloride ions are able to pass into the lumen of the respiratory tract to draw water out. Next guest, please introduce yourself. Hey, I'm a trafficking defect, the most common type associated with CF. What happens with me is that a misfolded nascent CFTR protein is created. So instead of continuing into the Golgi to be modified and sent to the cell membrane, the CFTR protein is marked for destruction. No. I too result in no chloride ions being able to leave the cell. Next guest, the floor is yours. I hardly need any introduction, but I'm defective channel regulation. Everything happens pretty much normally: transcription, translation, trafficking. Most part, it's good. The real issue is when the CFTR does not properly allow the transport of chloride ions. So the chloride ions just sit inside the cell and are not able to get out. That's the main problem. Next guest, please. Yo, I'm defective CFTR protein. A DNA mutation results in a formation of a protein that has a reduced channel conductance. The defective protein doesn't let out the right amount of chloride ions, resulting in a small amount of chloride ions present in the lumen and very little water being drawn out the cell. No. Next guest. Hello everyone, I'm scarce functional CFTR. Sometimes the RNA is incorrectly transcribed and will not give rise to a functional CFTR protein. Due to the low amount of CFTR present in the apical membrane, the cell is totally unable to pump a sufficient amount of chloride ions into the lumen, which draws water out of the cell. Last but not least, hey, I'm decreased CFTR membrane stability. A mutation in the DNA causes the expressed CFTR protein to be unstable in the cell's membrane. After the protein is trafficked to the membrane, it is able to perform its function as an ion channel. However, due to its defect, it may slip out of the membrane. This results in low amounts of CFTR on the cell membrane. Well, that's all we have time for today, folks. See you next time on CFTR Protein Defect Red Table Talk.